Good morning, students. In today's class, I am going to continue the second topic, biological classification, with kingdom Monera. So, in today's class, we will discuss about the kingdom Monera. So, all prokaryotes have placed in the kingdom Monera. Exclusively, all prokaryotes included in kingdom Monera. Kingdom Monera has divided into two groups. Divided into two groups. One group is called the Archaebacteria. Another group is called the Eubacteria. Archaebacteria is also called the primitive bacteria, whereas Eubacteria are called the true or advanced. They are called the true or advanced. Archaebacteria again divided into three categories: halophils, methanogens, and thermophils. In the same way, bacteria also divided into two groups. One is bacteria and Cyanobacteria. So first, we will discuss about the Archaebacteria. As we know, Archaebacteria are primitive bacteria, and these Archaebacteria survive in very harsh conditions or extreme conditions where other organism survival is very difficult. The harsh they survive in harsh habitats like salty area, marshy area, and in hot springs. These are some of the extreme conditions or extreme habitats. So these bacteria can survive in such a harsh habitats. And they can survive in such a harsh habitats because of their cell wall. Because they have a unique cell wall, and this unique cell wall will help them to survive in such a harsh condition. So the Archaebacteria are divided into three types based on their habitats. If the Archaebacteria survive in salty area, they are called the halophils. Example, Halococcus. If the Archaebacteria survive in marshy area, they are called the methanogens. If the Archaebacteria survive in the hot springs, they are called the thermophils. Example for methanogens: methanococcus, and example for thermophils are thermoprotease. So Archaebacteria are primitive bacteria. They survive in the harsh habitats like salty area, marshy area, hot springs. These are very harsh habitats where other organisms cannot survive. They can live in such harsh condition because of their cell wall, because they have very unique cell wall. The definition of three types of Archaebacteria. Archaebacteria which survive in salty area are called halophils, example Halococcus, and Archaebacteria which survive in marshy area are called the methanogens, example Methanococcus, and those which survive in hot springs are called the thermophils. Let us continue the next type of monerans. Eubacteria. Eubacteria. Eubacteria are true bacteria. These are true bacteria. They have rigid cell wall. Rigid cell wall made by murine or peptidoglycan. Peptidoglycan. Eubacteria are true bacteria. They have rigid cell wall and which is made by chemically murine or peptidoglycan. The cell wall is surrounded by a gelatinous sheet. The external surface of the cell wall is covered by a gelatinous sheet which is called the glycocalyx. The surface of the cell wall is covered by a gelatinous layer and that layer is called the glycocalyx. Glycocalyx can then be differentiated into two types. We may call it as a slime layer or capsule. Glycocalyx may be slime layer or capsule. Slime layer is the one which is smooth, loose sheet made by polysaccharides. Whereas capsule is hard or rough and it's made by polysaccharide and polypeptides. They may be motile or non-motile. If they are motile, they have flagella as a locomotory structure. Eubacteria can be divided into two types. Types of Eubacteria. One is bacteria, another one is Cyanobacteria. Let us first discuss about the bacteria. So based on the structure, bacteria divided into 4 groups. There are 4 groups. Shade, we may call this shade. based on the shape of the organism. Bacteria, 4 types of bacteria are there. First one is PUC-I or singular coccus, means spherical shape. If the bacteria are spherical shape, they are called the coccus or PUC-I. Shape of the organism is like this. They may also call the bacilli. The bacteria are rod shaped. If they are rod shaped, then they are called the bacilli or singular bacillus. Next type is spirular. Spiral shaped bacteria are called the spiral, or called the spiral shaped or called the spiral. Fourth one is vibrio. Comma shaped bacteria are called the vibrio. So like this, bacteria can be divided into 4 types based on their shapes. Next, bacteria chemosynthetically autotrophs, or they may be heterotrophs. Chemosynthetically autotrophs means they can get their energy by the oxidation of certain chemicals. They get energy by oxidation of certain chemicals like nitrite, nitrate, ferrous, sulfur, etc. Example for these chemosynthetic bacteria: Rhizobium, Nitrosomonas, Nitrobacter. Then heterotrophs. Heterotrophs are those which gets their food from other organisms, and they may again divided into 3 categories: saprophytes or decomposers. Parasites or symbiotic bacteria. So heterotrophs may be 3 types. Next is reproduction in bacteria. Reproduction in bacteria. Bacteria express 2 types of reproduction. One is sexuality or sexual reproduction, and another one is asexual type of reproduction. Asexual reproduction takes place by the help of 2 structures or 2 types. One is by fission, another one is by endoscopes. In fission, whenever a bacteria divides into 2 daughter cells, that we call the fission. Endoscopes usually develops during unfavorable unfavorable conditions. When the conditions are normal, fission is a common type of asexual reproduction. When conditions are adverse or unfavorable, bacteria develops the endospores. Means the whole content of the bacteria develops a thick wall to protect the inner content. Once the normal conditions come back, so this thick wall degenerates and again it starts producing as usual asexual reproduction. Normal asexual reproduction. In parasexuality, parasexual reproduction means introduction of new genetic type or combination in organisms without gametogenesis and fertilization. Parasexual means introduction of new genetic character without gametogenesis and fertilization. Examples for this: conjugation, transformation, and transduction. These are the three types of parasexual reproduction. Bacteria may be useful or harmful. They may be useful. Useful bacteria. Some bacteria may be useful and some may be harmful. Useful bacteria are those which help in the production of many things. For example, in the preparation of curd, antibiotics, organic acids, bioactive molecules or bioactive substance, and also they use as a biofertilizers. So, bacteria which help in this way are called the useful bacteria. And there are some bacteria which are harmful. Harmful bacteria. Harmful bacteria are those which cause the damage or which may cause the disease to the plants, animals and even human beings. Some bacteria may damage the packed food, that we call the botulism. And they may also cause the disease to the plants as well as animals, including human beings. Example for disease: citrus canker. Citrus canker, which cause the disease to the citrus plant. And also many diseases to the human being: typhoid, pneumonia, tetanus, cholera. These are some of the bacterial diseases to the human being. So next we discuss about the Cyanobacteria. We'll continue the another eubacteria, Cyanobacteria. Cyanobacteria is also called the blue-green algae. They are also called the blue-green algae. These cyanobacteria may exist in unicellular form, colonial form or filamentous form, and their colonies are surrounded with gelatinous layer. That gelatinous layer is called the mucilage. Gelatinous layer which cover their colony is called the mucilaginous layer. They may be marine or terrestrial. They may be marine or terrestrial form. Terrestrial forms. Usually cyanobacteria photosynthetically autotrophs. These are photosynthetically autotrophs. Because they have the chlorophyll A, and by using chlorophyll they get their energy by process photosynthesis like other plants. They get their energy by photosynthetic process like other plants. And some of the cyanobacteria like Nostoc and Anabaena are very useful in atmospheric nitrogen fixation. They fix the atmospheric nitrogen because in their filament there is a specialized cell called the heterocyst. Heterocyst is a specialized cell in their filament which fix the atmospheric nitrogen. They may fix the atmospheric nitrogen in free state or in symbiotic form. Nostoc fix the atmospheric nitrogen along with the coralline roots of cycas in symbiotic relation. In the same way, Anabaena fix nitrogen, atmospheric nitrogen in a symbiotic relation with the azote. And last part is mycoplasma. Mycoplasma are the smallest cells, smallest living cell. Living cell, they lack or without cell wall. Without cell wall and survive in absence of oxygen. Survive without oxygen. And they can cause many diseases to the animals and plants. So here I conclude kingdom Monera. In the next class we will continue the next kingdom, kingdom Protista. Thank you.